Good afternoon, all. Thanks all for having me today. This is a great honor for me to be invited to this conference. Pseudomonas aeruginosa is classified as a priority 1 pathogen by the World Health Organization due to the emergence of multi-drug resistant strains. This afternoon, I will try to explain the study that we did in my country in the Niño Hospital and how the virulent factors are important in the prevalence of antibiotic resistance in Pseudomonas aeruginosa. Pseudomonas aeruginosa is a gram-negative aerobic rock-shaped bacterium that belongs to the gamma subclass of protobacteria and the rRNA group 1 that can be divided into non-fluorescent and fluorescent. This is an ubiquitous bacterium that can colonize multiple niches and utilize many environmental compounds as an energy source. As a human pathogen, it is a prevalent opportunistic pathogen. It's responsible for nosocomial infections due to the use of medical appliances such as urinary catheters, respirators, venous access. The use of contact lens as well can produce eye infections. In immunocompromised patients such as cystic fibrosis, AIDS, born patients, organ transplant recipients, is responsible for the high rate of illness and death. The capacity of Pseudomonas aeruginosa to produce such overwhelming infection is due to their arsenal of virulent factors and that are responsible for the multi-drug resistant strains. The multi-drug resistant and thus the bacterium virulence depends on a large number of cell-associated and extracellular virulent factors. As we see here, the cell-associated are the pili, the flagellum, the lipopolysaccharides, outer membranes proteins such as porins, lipoproteins, secretion system, iron uptake system, alginate as a part of the biofilm. Extracellular virulent factors are exotoxin, proteases, lipases, elastases, pyoverdine siderophore as a part of the iron uptake system. The role of these virulent factors are important for cell colonizations, bacteria survivals, and tissue invasions. It is important as well to understand how the antibiotics are uptake in Pseudomonas aeruginosa. For example, macrolides are diffused through the lipid bilayer because of their hydrophobic natures. Once inside, they bind to the 58 ribosomal subunit and inhibit protein synthesis. Aminoglycosides diffuses in the outer membrane layer because of the interactions between the lipopolysaccharides and the aminoglycosides. They diffuse in an ATP-dependent manner and bind to the 38 ribosomal subunit. Quinolones and beta-lactams use porins. Once inside, quinolones target the gyrase 4 and topo-4 polymerase and inhibit DNA replication. Beta-lactams inhibit penicillin-binding proteins and therefore cell wall synthesis. The resistant mechanism of Pseudomonas aeruginosa is divided in three types. The intrinsic resistant mechanisms are those genetically encoded in the core genome of Pseudomonas aeruginosa. Acquired resistance arise by the gain of de novo genes or from other organisms or as a selection of an advantageous mutation. Adaptive results from the induction of environmental stimuli. As we see in the figure, the bacteria fights against antibiotics in several ways. For example, enzymatic degradation, alteration of drug binding target, loss of drug entrance. But the most common form of resistance to most classes of antibiotics is through pumping out antibiotics of these cells. Pseudomonas aeruginosa has important efflux systems for this purpose. The bacteria multidrug efflux systems can be classified in two larger superfamily, the major facilitator superfamily, the ATP binding cassette superfamily, and three smaller families, which are the small multidrug resistant, the multidrug and toxic compound extrusions family. But the most important for gram-negative bacteria are the resistant nodulation cell division family, and Pseudomonas aeruginosa, as we see here, has several efflux systems that make it resistant to most antibiotics. The MEX-AB-OPRM efflux system is the major POM in Pseudomonas aeruginosa that contributes to the clinical antibiotic resistance. It is composed of three proteins, one outer membrane protein, OPRM, one perisplasmic protein, MEX-A, and one inner protein, MEX-B. This protein has important domains. The inner membrane proteins has domain that provide the energy as a proton-motif form. It has a pore domain that has gates for the uptake and extrusion of the drug, and has a docking domain that connects to the OPRN. MEX-A is the fusion protein. It means that it fuses the outer membrane protein and the inner membrane protein. And the outer membrane protein, OPRM, has a alpha helices domains that connect to the fusion protein and the beta barrel domain. MEX-AB-OPRM operon is transcriptionally regulated to three ways. The main repressor is MEX-R, that due to the reactive oxygen species no longer bind to the operator, and the MEX-AB operon is activated. The transcription is activated. The binding of pentachlorophenol to NAL-C that represses the ARM-R, and it binds to the MEX-R repressor, and no longer the MEX-R repressor controls the transcription. The NAL-D is the secondary repressor and has been shown that when it interacts with the antibiotic novobiosyn, the operon is de-repressed. Another important antibiotic resistant mechanism are beta-lactams. As I mentioned, beta-lactams blocks the cell wall synthesis by inhibiting the penicillin binding proteins. The cell wall is composed of two amino sugars, a peptide chain, and a cross-link. The penicillin binding proteins catalyze the last reaction in this process by the hydrolysis of two contiguous dealanine residues. Therefore, they are responsible for the cross-link between contiguous peptides. But penicillin mimics these two last residues, and the enzyme PBP cannot identify it, cannot differentiate between the dealanine residues and penicillin, and therefore the inhibition of cross-linking occurs. A type of PBP genes exist in the genome of Pseudomonas aeruginosa, 3 chromosomal beta-lactamase, class A, class D, class C, and 5 high molecular weight PVP that has a domain for glycosylase and transpeptidase activities. 3 other proteins are the protein 5, the protein 7, and protein 4, which are carboxypeptidases that are widely resistant to various beta-lactams. Well, as we see in this figure, if we compare the action of the beta-lactams with the penicillin binding proteins, they break the same covalent bonds. So it is suggested that the beta-lactams evolutionate from the penicillin binding proteins. As we see, the target for beta-lactamase is the beta-lactam ring. Here we observe other antibiotics that has the beta-lactam rings. Lactamases can be divided into two main groups depending on how they perform the hydrolysis of the beta-lactam ring. Beta-lactamases can be performed either by forming an acyl enzyme with an active setting state or by hydrolysis using a zinc ion in the active sites. So we see here serine beta-lactamase. It means that it has serine in its active site and is divided into 3 classes: Class C, Class A, Class C, and Class D. Among Class A, we have these beta-lactamases, CHB, TEM, and and among the Class C, the AMP-C, which were the subjects or the genes that we studied. Metal of beta-lactamase has zincs in the active site and correspond to the Class B. Well, the next group of genes that we select to study the resistance profile in Pseudomonas aeruginosa are related to the iron uptake. Iron is an essential element for almost all living organisms due to the redox potential that makes iron extremely versatile when it is incorporated into proteins as a catalytic center or as an electron carrier. Therefore, it is an essential element for nucleic acid synthesis, gene regulation, respiration, and photosynthesis. Iron has a low solubility in aerobic environments because it exists as a ferrine form that precipitates as an insoluble oxyhydroxy polymers, and therefore the concentration is too low for the microbial survival. Excess of iron is toxic for the cell, is a central component in the Haber-Weisfenton chemistry that produces reactive oxygen species, and therefore the concentration of iron must be regulated to avoid these toxic effects. What are the sources of iron in Pseudomonas aeruginosa? Iron has to uptake a system for M encoded by the Fu and has lossy. Iron are obtained as well from the M group, from the hemolyzing phospholipase C, and exotoxin A also uses the M group from hemoglobin and produces tissue necrosis, Multi-copper oxidase as well is involved in the uptake of ferrous iron, and the most important system is the siderophore. The siderophore of Pseudomonas aeruginosa are iron-chelated molecules of low molecular weight. Two important siderophores are the pyokelin, who has low affinity for iron, and pyoverdine, which has a high affinity for iron. This is a virulent factor that is acquired that acquire iron from the host transferrin. It has been reported as well as a plant growth promoter molecules. The pyoverdine is composed of a peptide chain, a variable peptide chain, a chromophore, which is responsible for the fluorescence and has a catechol group that is responsible for iron uptake, and an acyl chain. The pyoverdine biosynthetics genes are located at the PBD locus and expands from PBDQ to PBDS genes in the sequence genomes of Pseudomonas aeruginosa. Three structurally different proteins has been sequenced that are recognized by its own cognate receptor, which are the type 2, type 1, and type 3. FPBA receptor belongs to the TOMB dependent receptor. It has several domains. It has a FPBR box, a TOMB box, a plug domain, and a barrier domain. As we see in this figure, we observe how the conformation of the pyoverdine receptors change when the ligand bind or not bind. For example, this is the conformation when there is no ligand, and here we see that the plug domain changes in conformation, especially the switch helix. In the beta barrier domain, two alpha helices, the LA and L7 as well, changes in conformation. How this transport occur of the siderophores? The ferry siderophore are too large to diffuse through the porins in the outer membrane of the bacterium. So they need a TOMB XBXD complex that provides the energy for the binding to the receptor. Once they bind to the receptor, they are transported to the periplasmic ATP binding proteins that depends on the hydrolysis of ATP, and they traverses the inner membrane foci, and inside the cytoplasm there is a reductase that causes the reduction of ferric iron. The pyoverdine synthesis and transport is regulated. They have a global regulator through the full repressor that does not bind to the biosynthetic genes or the receptors genes. Instead, it binds to the promoter region of 2 sigma factors, and therefore, when there is no pyoverdine, the transcription is not activated. The pyoverdine itself, the ferric pyoverdine itself, regulates the transcription of the genes via an anti-sigma factor, FPBR, that in the absence of ferric pyoverdine binds the sigma factor. When pyoverdin is present, it causes the FPBR to release the sigma factor and the transcription of the genes occurs. Which were the aims of our research? Well, we wanted to characterize the antibiotic resistant profile of 51 clinical isolates of Pseudomonas aeruginosa obtaining from children admitted to the Hospital del Niño Panamá. We wanted to characterize if the isolates produces pyoverdin and if these isolates pyoverdin produces were related to the expression of the MEX-AB OPRM operon, the pyoverdin receptor, and the presence of the beta-lactamases, BLATEM, BLAST-HAB, and BLAST-CTX-like. As we see in this figure, we observe the resistance profile of the different antibiotics of the 51 isolates. As we see here, the bacteria were more sensitive to aminoglycosides, fluoroquinolones, and cephalosporin, a kind of beta-lactamase. And they were more resistant to beta-lactams, as we see 69%, 35 non-resistant isolates, followed by metamines and nitroforans antibiotics. We proceed with the molecular detection of the MEX-A, MEX-B, MEX-R efflux systems, and we identify amplicons for the 3 genes in pyoverdin isolates. So it means that the pyoverdin favor the expression of these genes. We detect as well the pyoverdin receptors, and we see different profiles for each of the 3 receptors. We observe 44% of the FPBA1 type, 35% of the FPBA3 type, and 31% of the FPBA2 type, meaning that the production of pyoverdin increases the virulence of the bacterium. Finally, we detect the BLATEM-like beta-lactamases genes, and the pyoverdin pigment production is ubiquitous between these beta-lactamases strains of Pseudomonas aeruginosa, because pyoverdin and beta-lactamase production are necessary for the acquisition of drug-resistant capabilities. What are the concluding remarks? Pyoverdin is a virulent factor necessary for the expression of the genes that we study, the FPBA receptor genes, the MEX-AB OPRM efflux pumps, the BLATEM-like beta-lactamases. These virulent factors contribute to the antibiotic resistance of Pseudomonas aeruginosa. Pseudomonas aeruginosa has the capacity to produce a wide range of virulent factors, which make the multi-drug resistant pathogen for which there are limited therapeutic options. Therefore, the understanding of resistant mechanism can facilitate the development of new therapeutics. That's all. Grazie molte. Gracias.
Thank you very much.